Bismillah ar-Rahman ar-Rahim. Today is our second lecture in the course of biology as a part of continued education program. The big title of our lecture is Protein Synthesis. The objectives of this lecture is to understand the concept of nucleosides, nucleotides, and nucleic acids, what's meant by replication, including mitosis and meiosis, amino acids and protein structures, protein synthesis, catabolism of amino acids, metabolic functions of amino acids. We will start this lecture by understanding the concept of nucleosides, nucleotides, and nucleic acids. Nucleosides contain sugar linked to a nitrogen-containing base. The physiologically important bases that are included in the nucleoside structures are either purines or pyrimidines, and these structures have ring structures. They are bound to either ribose or to deoxyribose to complete the nucleoside. When inorganic phosphate is added to the nucleoside, nucleotide is formed. So, nucleotide equals nucleoside plus inorganic phosphate. This is purine nucleus. This is pyrimidine nucleus. The forms of purine compounds, either adenine, guanine. The forms of pyrimidine, cytosine or uracil or thymine. Nucleosides and nucleotides form the backbone for RNA and DNA, as well as a variety of coenzymes and regulatory molecules like NAD, NADP, and ATP. These compounds have a physiological importance to every cell in your body. We have other examples of purine and pyrimidine containing compounds. For example, for nucleosides, the component purine, pyrimidine plus ribose or 2-oxyribose nucleotides equals nucleosides plus phosphoric acid or inorganic phosphate. Nucleic acids, many nucleotides forming double helical structure of 2 polynucleotide chains. Nucleoproteins, which consist of nucleic acid plus 1 or more simple basic proteins. RNA, it contains ribose. DNA, which contain 2-deoxyribose. Nucleic acids in diet are digested and their constituents, purines and pyrimidines, absorbed, but most of the purines and pyrimidines are synthesized from amino acids, principally in the liver. The nucleotides and the RNA and DNA are then synthesized. RNA is in dynamic equilibrium with the amino acid pool in your body, but DNA, once formed, it is metabolically stable throughout life. So this is a big and very important difference between RNA and DNA. The idea of stability. So DNA is stable throughout life, metabolically stable throughout life, but DNA is in dynamic state. It is in dynamic equilibrium with the amino acids pool in your body. The purines and the pyrimidines released by the breakdown of nucleotides may be reused or catabolized. Small amount of these purines or pyrimidines are excreted unchanged in urine. The pyrimidines are catabolized to beta amino acids, beta alanine, and beta amino isobutyrate. These amino acids have the amino group on beta carbon, and this makes the difference between these beta amino acids and alpha amino acids. So it is the orientation of the amino group either to the beta carbon or to the alpha carbon, which determine if this amino acid is alpha amino acid or beta amino acid. So for purines, they are catabolized to beta amino acids. Because beta amino isobutyrate is the product of thymine degradation, it can serve as a measure of DNA turnover. So chemically, and or clinically or laboratory, we can measure beta amino isobutyrate as a measure for DNA turnover. The amino acids are further degraded to carbon dioxide and ammonia. Uric acid is formed by the breakdown of purines and by direct synthesis from 5-phosphoribosyl pyrophosphate and glutamine. Here, this figure illustrates the process of synthesis and breakdown of uric acid. As you can see here, adenosine is converted to hypoxanthine, and then hypoxanthine is converted to xanthine by the action or by the activity of the enzyme xanthine oxidase. Then xanthine is converted to uric acid, again a reaction catalyzed by the same enzyme xanthine oxidase. Guanosine is converted directly to xanthine. Yeah, it's clear here in this image. Also, another reaction between glutamine and 5-BRPB is converted directly to uric acid. The combination between both is converted directly to uric acid, and then this uric acid is excreted in humans. But other mammals, this uric acid may be converted to allantoin. So in in human, uric acid is excreted in urine, but in other mammals, uric acid is further oxidized to allantoin before excretion. The normal uric acid level in human is about 4 mg per deciliter. In the kidney, uric acid is filtered, reabsorbed, and secreted. So renal handling of uric acid includes filtration, reabsorption, and secretion. Normally, about 98% of the filtered uric acid is reabsorbed, and the remaining 2% makes up approximately 20% of the amount excreted. The remaining 80% comes from the tubular secretion. So uric acid excretion on a purine-free diet is about 0.5 gram for over 24 hours, and on a regular diet about 1 gram over 24 hours. Excess uric acid in blood or urine is a characteristic of gout. So here we have a clinical application on uric acid excretion. A common clinical finding, or you can see cases of have this condition, which is called gout. Gout is a disease characterized by recurrent attacks of arthritis, urate deposits in the joints, kidneys, and other tissues, and elevated blood and uric acid levels. The joint most commonly affected initially is the metatarsopharyngeal joint of the great toe. This is affection of this joint by urate crystals, the first and the most commonly affected joint. There are two forms of primary gout. So for the etiological classification for the causes of gout, we can classify it either to primary gout or secondary gout. And then primary gout is further divided into two causes. The first form of primary gout, uric acid production is increased because of different enzyme abnormalities. The other form of primary gout, there is selective deficiency in renal tubular transport of uric acid. So two causes can lead to primary gout. Either increased production of uric acid or defect in renal tubular transport of uric acid. The other big type of gout is secondary gout. Here the uric acid levels in the body fluids are elevated due to decreased excretion or increased production secondary to some other disease process. So it is somehow indirect process leads to accumulation of uric acid. it is not the defect is not in the process of uric acid synthesis or not in the renal handling. For example, production is increased in leukemia and in pneumonia because of increased breakdown of uric acid rich white blood cells. The treatment of gout is aimed at relieving the acute arthritis with drugs such as colchicine or non-steroidal anti-inflammatory drugs and decrease uric acid level in blood. Colchicine is very well-known drug used in gout. It doesn't affect uric acid metabolism and it apparently relieves gout attacks by inhibiting phagocytosis of uric acid crystals by white blood cells, and this process in somehow reduce the joint symptom. Phenylbutazone and propanseed inhibit uric acid reabsorption in the renal tubules. These are other two drugs can be used in treatment or in management of gout. Allopurinol, this drug directly inhibits xanthine oxidase enzyme in the process of uric acid formation, and it is one of the drugs used to decrease uric acid production. Now we are going to talk about DNA structure. DNA, deoxyribonucleic acid, it is found in bacteria and in the nuclei of eukaryotic cells and as well as in the mitochondria. It's made up of two extremely long nucleotide chains containing the bases adenine, guanine, thymine, and cytosine. And as you can see here, every one of these bases is abbreviated into a symbol. Adenine abbreviated as A, guanine as G, thymine as T, and cytosine as C. This is the structure of typical deoxyribonucleotide, and this is the structure of typical ribonucleotide. The main difference that you can see here is the sugar. The sugar in typical deoxyribonucleotide is deoxyribose, and the sugar in typical ribonucleotide is ribose. Here, the structure of the bases for adenine can be found in both DNA and RNA. Guanine can be found in DNA and RNA. Cytosine in DNA and RNA. But here, thymine is present only in DNA and uracil is present only in RNA. The chains of DNA molecule are bound together by hydrogen bonds between the bases, with adenine bound to thymine and guanine bound to cytosine. This stable association forms double helical structure of DNA molecule. You can see here double helical structures and the combination between two bases. This is hydrogen bonds between T and A, C and G, A and T, and so on. This is the length of here 3.4 and this is the width 2 nanometer. The double helical structure of DNA is compacted in the cell by association with histones and further compacted into chromosomes. So to include the strand of DNA inside the small nucleus, it has to be compacted, it must decrease in size, and this compact structure or this decrease in size can only occur or can only be achieved through combining the DNA molecules to histones, and then these histones are compacted into chromosomes. The human cell contains a diploid number of chromosomes which equals 46 chromosomes. So this is DNA molecule and this is histone. It's combined with histone and then compacted again into chromosomes to be able to include it inside the nucleus. A fundamental unit of DNA or a gene, which can be defined as sequence of DNA nucleotides that contains the information for the production of an ordered amino acid sequence for a single polypeptide chain. So again, once again, this is the DNA structure, and this is a chromosome. Here, this segment is the gene, and this segment code for the formation of single amino acid. So it contains the information for the production for a single polypeptide chain, not single amino acid. So it gives the code for synthesis of certain polypeptide chain with an ordered amino acid sequence. The protein encoded by single gene may be subsequently divided into several different physiologically active proteins, because as we are going to see in this lecture, after protein synthesis, or after encoding what is present in this gene and transform it into polypeptide chain, the following step is post-translational modification. So a single gene may give rise to different physiologically active proteins by post-translational modification. In eukaryocytes, the portion of genes that cause the formation of proteins are usually broken into several segments. So gene is divided into several segments, divided into main partitions, some called exons and others called introns. Exons and they are separated by segments that are exons are the parts that are going to be translated, and they are separated by introns, and these introns are not translated. Actually, the main function of intron is unknown. So exons is going to be translated to give rise to the polypeptide chain that we need to synthesize, but introns are not translated, but they are present at intervals from exons. So this is the structure of the genes, alternating segments of exons and introns. Exon 1, intron 1, exon 2, intron 2, exon 3, and so on. So these exons are the parts that are going to be translated to form the target polypeptide chain. And these introns are not translated. These are intervening segments. So again, this is the gene with the gene which is a part of DNA that code for the synthesis of certain polypeptide chain. And here, clear it's the structure of introns and exons, or exons and introns. The exons for the transcription, or to form this polypeptide chain, we have two major stages. The first one called transcription. Transcription means synthesis of RNA from DNA, and then after RNA is formed, and RNA to participate in protein synthesis, it has to pass or had to move from the nucleus to the cytoplasm. And before moving from the nucleus to the cytoplasm, a process called RNA splicing occurs. RNA splicing means we remove these intervening introns that are not part of the transcription of the encoding. As we previously said, these introns are not expressed, so the rule is unknown. What have a physiological importance for me to synthesize the polypeptide chain is exon, not the intron. So in the process of RNA splicing, intron segments are removed, and then exons just come in close to each others, and these are the parts that will give rise after encoding to the polypeptide chain or to the protein in the process called, or in the other major stage of protein synthesis, which is called translation. In translation, the code in RNA is transformed into proteins. Near the transcription, start site of the gene, there is a promoter. A promoter is the site at which RNA polymerase and its cofactors bind. So, in the gene, there is transcription site, and near this transcription site, there is another site called a promoter. A promoter is the place where RNA polymerase and its cofactors bind. These promoters include thymine, adenine, thymidine, adenine sequence, or it's called theta sequence or theta box. So, at the promoter where RNA polymerase bind, there is a sequence, certain fixed sequence called TATA sequence. It contains thymidine, adenine, thymidine, adenine. It's called TATA sequence or theta box. This promoter ensures that transcription starts at the proper point. So it is like a landmark. You can, when you are traveling somewhere from between cities, you see a landmark or a milestone on the way that you are on the right track. And for the process of protein synthesis, theta sequence or theta box ensures that transcription starts at the proper point, and RNA polymerase will bind at the proper point to start the process of protein synthesis. As you can see here, this figure illustrates the structure of the gene with introns and exons. Of course, introns will be spliced during the process of protein synthesis, and exons will be encoded. Here you can see promoters area and TATA sequence, which helps the site of binding for RNA polymerase. Further out in 5' region of DNA molecule, there are regulatory elements which include enhancers and silencer sequences. For every gene, there is about 5 regulatory sites. Sometimes the regulatory sequences are not present in 5' region, but instead in 3' area or 3' region. So this DNA molecules 5/end and 3/end. Here near the 5/end there is a regulatory region where there is enhancers or silencers for the process of protein synthesis, and here the promoter site or where you can see TATA sequence or TATA box, and here's the transcription start site. There is exons and introns as well. A very important concept that you should be familiar with is the meaning of gene mutations. Gene mutations occur when base sequence in DNA is altered from its original sequence. This alteration will affect protein structure and may pass to daughter cells after cell division. Here you can see G is changed into A, and here T is changed into G. So this is gene mutation or alternation in base sequences. Point mutations occurs if single base is substituted or altered. There is a number of or variety of risk factors that may lead to this condition of gene mutation, like alkylating agents, ionizing radiation, and these agents or these risk factors may lead to change in DNA sequence and occurrence of mutations. This single point mutation, where there is substitution of single base, replacement of single base on DNA molecule. The collection of genes within the full expression of DNA from an organism is termed the genome. So, the whole DNA inside the organism is called the genome, but the gene is the small part of DNA that encode for certain polypeptide chain. Please don't confuse between these definitions, the gene and the genome. So, genome and here for gene is only small part on DNA molecule or segment on DNA molecule that encode for the synthesis of certain polypeptide chain, or give the code for the sequence of certain amino acids to form specific or certain polypeptide chain. An indication of the complexity of DNA in human haploid genome is its size. It's made up of 3 times 109 base pairs that can code for approximately 30,000 genes. And this genetic message is the blueprint for the characteristics that's transferred between cells and its descendants. The proteins formed from DNA blueprint include all the enzymes, and this in turn controls the metabolism of the cell. That is why if you hear about certain gene mutations, you can find that there is a problem in metabolism for carbohydrates. You may find inborn errors of metabolism in lipids, in proteins, and so on, because these mutations affected these enzymes that control certain metabolic pathways in the process of carbohydrates, in the process of proteins, or in the process of lipid metabolism. Each nucleated somatic cell in the body contains the full genetic message, but there is great differentiation and specialization in the function of different types of adult cells. So only small part of this message is normally transcribed, and in turn this genetic message is maintained in a repressed state. So every cell has the whole message, but not every cell translates this full message. They only translate what the cell needs for its function. So the message remains in repressed state, and which part of the message is going to be repressed and which part is going to be expressed depend on the function and depend on specialization of this cell. Now we are going to move to another topic in today's lecture, which is cell replication, including mitosis and meiosis. At the time of each somatic cell division, the 2 DNA chains are separated, and each one of them serves as a template for the synthesis of a new complementary chain. And this reaction is catalyzed by an enzyme called DNA polymerase. So this is at the time of replication. We have this DNA in the normal form of double helical structure, and as you can see here, the 2 strands first separated here. At this point, this one is separated in one direction and the other chain is separated in another direction, and then each one acts as a template for the formation of another strand. So we have at the end of the process 2 strands of DNA. Each one is composed of two strands, or each one is double helical structure. So first, in the process of cell replication, DNA is copied, and this is mitosis occurs, in which DNA is split equally into 2 daughter cells. But these 2 daughter cells at this stage remain attached at this point, and to be separated into 2 individual cells and remove this part of connection or this connection, this process of complete separation is cytokine dependent. So under the effect of cytokines, parent cells is cleaved into halves, or we obtain 2 separate cells. One of the double helical strands of the DNA during the process of DNA replication goes to one daughter cell and one goes to the other. So the amount of DNA in each daughter cell is the same as that in the parent cell. The life cycle of the cell that begins after mitosis is highly regulated, and it is termed the cell cycle. Phases of the cell cycle. First, G1 or gap 1 phase. This phase represents the period of cell growth and divides the end of mitosis from the DNA synthesis, or S phase. Following DNA synthesis, the cell enters another period of cell growth, which is called gap 2 or G2 phase. The ending of this stage, or the ending of G2 stage, is marked by chromosome condensation and the beginning of mitosis, or M stage. So this is the stages of cell cycle. We have G1, in which cellular contents excluding chromosomes are duplicated, and then S, in which each of the 46 chromosomes is duplicated by the cell, and then G2, in which the cell doubles, checks the duplicated chromosomes for error, making any needed repairs, and then M, or mitosis. And here, under the effect of cytokinases, the two cells are separated into completely separated cells. Each one has the same number of DNA as the mother cells, or the cell before cell division. Meiosis, on the other hand, occurs in germ cells. In germ cells, reduction division, or meiosis, occurs during the process of maturation. The net result is that one of each pair of chromosomes end up in each mature germ cell, and so each mature germ cell will contain half the amount of chromosomal material found in somatic cells. Therefore, when sperm unite with an ovum, the result is zygote with full number of chromosomes, or with full complement of DNA, half of which come from the father and the other half come from the mother. So this is meiosis. Start with the full number of DNA and end with every daughter cell have half number of the chromosomes. Here, mitosis, on the other hand, the daughter cells contain the same number as the parent cell, or the original cells from which mitosis started. The term ploidy is sometimes used to refer to the number of chromosomes in the cell. Normal resting diploid cells are euploid and become tetraploid just before division. Aneuploidy is the condition in which a cell contains other than haploid number of chromosomes, or an exact multiple of it, and this condition is common in cancerous cells. Now we are going to move to RNA. RNA strand is not double helical structure like DNA strand. On the other hand, RNA is single strand molecule, and the strands of DNA give rise to these RNA molecules in a process called transcription. So DNA serve as templates for RNA by lining up complementary bases for the formation of RNA, or for the formation of ribonucleic acids. RNA differs from DNA in being single-stranded, and also there is structure of the sugar. In DNA it is deoxyribose, and in RNA it is ribose, and also in bases. For example, RNA is present in uracil, while DNA contain thymine base. So three main differences. First, in number of strands. RNA is single, while DNA is double stranded, or double helical structure. In the structure of sugar molecule, in RNA it is ribose, and in DNA it is deoxyribose, and in base structure, for DNA it is thymine, and in RNA it is uracil. Here you can see the structure of RNA, and this is DNA. This is single strand, single helical structure, and here it is double helical structure. You can see here it is sugar. No, sugar is not clear here, but bases are clear. Here you can see uracil and RNA, and for DNA you can see thymine. The production of RNA from DNA is called transcription. There are several types of RNA. We have mRNA or messenger RNA. We have tRNA or transfer RNA. We have rRNA or ribosomal rRNA. And they are not only three, there are other forms of RNAs in the cell, but these are the three mentioned types are the major players in the process of protein synthesis. So they are not only three. Keep this in mind, please. Transcription is catalyzed by different forms of RNA polymerases. So formation of RNA from DNA is called transcription, and it is catalyzed by a group of enzymes called RNA polymerases. So please keep in mind. Duplication of DNA during the process of mitosis under the effect of DNA polymerases. But transcription, which is the synthesis of RNA from DNA, is by the effect of RNA polymerases. So don't mix both of them. Don't confuse between both polymerases. So this is DNA. And first, here the helical structure is interrupted here, and it acts as a code or a template for the formation of RNA molecule. And here it is template strand. This strand will be act as a template one for formation of RNA. And we add complementary bases to this template strand to form RNA strand. And again, this process under the effect of RNA polymerase. When suitably activated, transcription of genes into pre-mRNA start at the cap site and end about 20 bases beyond certain sequence called AATAA sequence. So this is part of DNA. This is the gene part, here introns and exons. For transcription or formation of RNA from DNA, first a molecule called pre-mRNA is formed, and then this pre-mRNA undergoes certain processing, a process called RNA processing, to form the mature mRNA. In this RNA processing, the introns are removed, and then the exon parts will be fused together to form the mature mRNA. Here at this end we can see something called poly A end at one end of pre-mRNA and also on the mature mRNA, and on the opposite side there is cap end. It's present in pre-mRNA and as well as immature mRNA after RNA processing by removal of introns. So RNA transcript is kept in the nucleus by addition of 7-methylguanosine triphosphate. So this cap, you can see here, this cap is composed of 7-methylguanosine triphosphate. This cap is composed of 7-methylguanosine triphosphate, and it's present at the 5' end. So this is 5' end of mRNA. This cap is important for proper binding to the ribosomes in the process of protein synthesis. So capping occurs in the nucleus by addition of this structure to enable mRNA to bind to ribosomes in the process of protein synthesis. Another structure at the other end of RNA molecule, which is poly A tail, It is about 100 bases of adenine, about 100 adenine bases. Here it's poly A in the pre-mRNA and in the mature RNA. So it is a tail of about 100 adenine bases. It is added to untranslated segment and at 3/end. It maintains the stability of mRNA. Let's come back to the figure again. So we have cap, important for binding of RNA to the ribosomes, and we have poly A tail, important for stability of RNA molecule. The pre-mRNA formed by capping, so this is the cap for to maintain or to help binding of RNA to ribosomes, and this is poly A tail at the 3/end, so this is 5/end and this is 3/end. The poly A tail is important for stability of the molecule. The pre-mRNA formed by capping and addition of poly A tail, then processed by elimination of introns, and once this post-translational modification is complete, mature mRNA is formed. So for processing, the introns have to be removed. This is the introns and it's removed, and now we have mature mRNA which is ready for being translated. Post-translational modification of pre-mRNA is regulated process where differential splicing can occur to form more than one mRNA from single pre-mRNA. Let's understand this process more. We have this part or this molecule of pre-mRNA before processing. If I remove these intervening parts of introns, this intron and this is another intron and this is the third intron, then I have one, two, three, four. I have 4 exons. These 4 exons give rise to, if I encode these, if I have the code or encode the amino acid sequence of these exons, I will have certain polypeptide chains. Let's say name this part exon 1, exon 2, 3, and 4. So if I add 1 after 2, after 3, after 4, and have transcription for this molecule, I will have a sequence of certain amino acids and in turn certain polypeptide chains. But what happens if I change the sequence? For example, I put 1, then 3, then 2, then 4. In this case, I will have another sequences of amino acids and in turn different polypeptide chain. Let's think about third condition. What happens if I start with 4, then 1, then 2, then 3? Then I have a third sequence of amino acids and in turn a third polypeptide chain, and so on. This is the idea. If you change the sequence of exons in the process of processing, you can have a big number of polypeptide chains with every time you change the sequence of these exons, and in turn you will change the sequence of the resultant amino acids and the structure of the net polypeptide chain. The introns of some genes are eliminated by what's called spliceosomes. Spliceosomes are complex units that made up of small RNAs and proteins. I have just said that we don't have only 3 types of RNAs. The 3 mentioned types are the 3 main players of protein synthesis, but we have inside the cell other forms of RNA, and this is one of these forms, which is called small RNAs. Small RNAs in combination with certain proteins form another structure or component in the cell called spliceosome, and this spliceosome may eliminate the introns of some genes. Other introns are eliminated by self-splicing, so the process of RNA processing or removal of introns achieved by 2 ways, either by spliceosomes or by self-splicing. Because of introns and splicing, more than one mRNA again can be formed from the same gene. So this is splicing, this is intron, this is exon, this is exon 1, another exon, and splicing, I want to remove this part, and here this part is removed by spliceosomes. Most forms of RNA in the cell are involved in translation or protein synthesis. In the cytoplasm, ribosomes provide a template for tRNA to deliver specific amino acid to the growing polypeptide chain based on a specific sequence present in mRNA. The mRNA molecules are smaller than DNA molecules, and each represents a transcript of a small segment of the DNA chain. For comparison, the molecules of tRNA contain only from 70 to 80 nitrogen bases compared to the hundreds present in mRNA and the billions present in DNA. Now we are moving to another topic in our today's lecture, which is amino acids and proteins. Amino acids form the basic building blocks for proteins, and this table give you different classifications of amino acids. Here we have amino acids with aliphatic side chains like alanine, valine, leucine, and we have sulfur containing amino acid like cysteine. We have hydroxyl substituted amino acids like serine. We have amino acids with aromatic ring side chains like phenylalanine, and so on. These amino acids are often referred to by their corresponding three layers or corresponding three letters or single letter abbreviations. So you can use abbreviations to instead of writing the full name of the amino acid. Here you can see for alanine we may use ALA or A, either first three letters or single letter. For valine, VAL or V. For leucine, LEU and L, and so on, all of them. Here, for example, histidine, either the first three letters, HIS or H. There are other important amino acids like ornithine, 5-hydroxytryptophan, L-dopa, taurine, thyroxine. These amino acids are physiologically important in our body, but they are not present in proteins. They don't share in structure of of proteins. So other important amino acids, physiologically important, like for example L-dopa, it is in CNS or thyroxine hormone. These are or thyroxine, but they are not found in proteins. In higher animals, the L isomers of the amino acids are the only naturally occurring forms in proteins. The L isomers of hormones, such as thyroxine, are much more active than the D isomers. The amino acids are acidic, neutral, or basic in reactions, depending on the relative proportions of free acidic or basic groups in the molecule. Some of the amino acids are nutritionally essential amino acids, that is, they must be obtained in diet because they cannot be made in the body. Arginine and histidine must be provided through diet during times of rapid growth or recovery from illness, and they are termed conditionally essential. So arginine and histidine are not essential all the times. So in certain times or in certain conditions, like rapid growth or recovery from diseases, they must be supplied in diet. So the name conditionally essential. All other amino acids are non-essential, that your body can synthesize them in amount sufficient to meet the metabolic requirements. The amino acid pool. Amino acid pool is a very important concept. Also, small amounts of proteins are absorbed from gastrointestinal tracts, and some peptides are also absorbed. Most ingested proteins are digested and their component of amino acids are absorbed. The body's own proteins are being in a continuous state of hydrolysis to amino acids and resynthesis. So there is a dynamic state. Our own proteins are not stable. They are in a dynamic state of breakdown and resynthesis. The turnover rate of endogenous proteins averages from 80 to 100 gram per day, being highest in intestinal mucosa and practically nil in extracellular structural protein collagen. The amino acids formed by endogenous protein breakdown are identical to those derived from ingested protein. Together they form a common amino acid pool that supplies the needs of the body. So this amino acid pool, as you can see, there is conversion or dynamic state between the amino acid pool and our body proteins. This amino acid pool receive its content from dietary sources, and as you can see here from the dynamic state or the dynamic exchange breakdown from the body proteins. So this amino acid pool can be participate in urinary excretion of amino acids, formation of creatinine, purine, pyrimidine, or formation of hormones and neurotransmitters. Between amino acid pool and common metabolic pool, there is certain metabolic reactions or transformations like transamination, amination, or deamination. The result of the process or the reaction of deamination is the formation of urea. So if you measured urea and creatinine or urinary content of amino acids, we can express the activity or the condition of this amino acid pool. Proteins are made up of a large number of amino acids connected together by peptide linkages or peptide bonds. These peptide bonds present between the amino group of one amino acid and the carboxyl group of the next amino acid. In addition, some proteins contain carbohydrates, and the result is formation of glycoproteins, or lipids, and the result is formation of lipoproteins. Smaller chains of amino acids are called peptides or polypeptides. So here you can see the peptide linkage. This is amino acid, and there is another amino acid or the next amino acid, and the wall is the polypeptide chain. Here the linkage is formed between carboxyl group of one amino acid and the amino group of the next amino acid. Amino acid chains contain from 2 to 10 amino acids molecules called peptides. Chains containing more than 10 but fewer than 100 amino acids are called polypeptides, and long chains containing 100 or more amino acids are called proteins. So the definitions of peptides, polypeptides, or proteins, they are the same ideas, combination of amino acids, but what makes the difference and make me call this peptide and this polypeptide and this protein is the number of amino acid that form that chain. From 2 to 10 it is peptide, from 10 to 99 it is polypeptide, from 100 and above it is protein. The order of amino acids in the peptide chain is called primary structure of proteins. The chains are then twisted or folded in complex way, and the result is formation of secondary structure of proteins. So secondary structure of proteins refers to special arrangement produced by twisting and folding of the chain. A common secondary structure is regular coil with 7.3 amino acid residue per turn, or alpha helix. Another common secondary structure is beta sheet. So we have primary, secondary, but not only primary and secondary, we also have tertiary structure of proteins, which is the arrangement of the twisted chains in two layers, crystals or fibers. Many protein molecules are made of several proteins or subunits, like hemoglobin, and then the term quaternary structure is used to refer to the arrangement of the subunits into the functional structure. So, you have to differentiate between primary structure, secondary structure, tertiary structure, and the final functional quaternary structure. Now we are moving to the very important process of protein synthesis. The process of protein synthesis or translation is the conversion of information encoded in mRNA to a protein. When certain mRNA reaches a ribosome in the cytoplasm, it guides the formation of polypeptide chain. Amino acids in the cytoplasm are activated by a combination with an enzyme, adenosine monophosphate, and each activated amino acid then combines with a specific molecule of tRNA. So, for any amino acid to participate in the formation of certain polypeptide chain as a part of the process of protein synthesis, this amino acid must be activated. It's activated by combination with ATP in the presence of specific enzymes, and then amino acid combined with AMP, and here it is activated amino acid. Here it can bind or it can be transferred on tRNA to join the process of protein synthesis. There is at least one tRNA for each of the 20 unmodified amino acids found in large quantities in the body proteins. But some amino acids have more than one tRNA. The tRNA amino acid adenylate complex is next attached to mRNA template in a process that occurs in the ribosomes. The tRNA recognizes the proper spot to attach on mRNA template because it has on its active end a set of 3 bases that are complementary to the set of 3 bases in a particular spot on mRNA chain. So there is complementation in the arrangement of bases on mRNA and on tRNA. Okay, so this complementary structure allow for proper attachment between both mRNA, which have the code for polypeptide chain synthesis, which has the decision what will be the arrangement of amino acids in this polypeptide chain, and this complementary structure will combine with its opposite structure or complementary basis on tRNA molecule, which carry the activated amino acid, which will join the forming polypeptide chain. The genetic code is made up of such triplet. So the codon or triplet of bases code for single amino acid of 3 sequence of 3 purines, pyrimidine, or purine and pyrimidine bases. So they are either 3 purines or 3 pyrimidines or combination of purine and pyrimidine. Each codon stands for a particular amino acid. So this is DNA molecule. This is the start of the story. DNA molecule with part of it for the gene, and then from DNA we have mRNA transcription in the process previously described in our lecture. And here the one of the DNA strands act as a template, and in presence of RNA polymerase enzyme, complementary bases are added to form pre-mRNA, which undergo certain processing and splicing, either by spliceosomes or self-splicing, to form the mature mRNA. Now, mature mRNA with only exons, as in the process of processing introns have been removed, can move through nuclear pores. This is a nuclear pores. It can pass through nuclear pores to the cytoplasm, where it will start the process of translation. So here's the process of translation is the formation of polypeptide chain according to the information present on mRNA. This is mRNA, and here the site of formation is ribosomes, and this is amino acid, and this is tRNA, transfer RNA, which carries the amino acid. And for amino acid to activate, to participate in the process of protein synthesis, or to join the polypeptide chain that is currently forming, or we are currently forming, it must be activated. This is amino acid protein, the developing polypeptide chain in the process. Translation typically starts in the ribosome when certain arrangement called AUG. This AUG or this codon code for methionine amino acid. AUG here it is present on the mRNA, but it is transcribed from ATG, the complementary ATG in the gene. It's transcribed from ATG in the gene, which and this codes for or this arrangement of bases codes for methionine. The amino terminal amino acid is then added, and the chain is lengthened one amino acid at a time. So at any time I can only add one amino acid and then the other, and so on. The mRNA attaches to 40S subunit of the ribosome during the process of protein synthesis. For your information, the ribosomes consist of two subunits, 40S and 60S, and here mRNA combine or attaches to 40S subunit. And the polypeptide chain, on the other hand, attached to 60S subunit. For tRNA, it combined to both of them. Let's see if it is clear on the figure or not. Now the subunits are not clear here. So this is the ribosomes. It contains, as an organelle, consists of two subunits, 36 and 40S subunits, 40S and 60S, and for mRNA it combined with 40S, and for the formed chain it combined with the heavy one, 60S, and for tRNA it attaches to both. As amino acids are added in order dedicated by the codon on mRNA, the ribosome move along mRNA molecule like a bead on a string. And then translation stops at one of three stop or nonsense codons. So when we reach these codons, the process stops. The stop codons either UGA or UAA or UAG. And then the polypeptide chain is released from the ribosomes. The tRNA molecules are used again. The mRNA molecules are typically reused approximately 10 times before being replaced. It is common to have more than one ribosome on a given mRNA chain at a time. The mRNA chain, with its collection of ribosomes, is visible under electron microscope as an aggregation of ribosomes called polyribosomes. So this is the imaging by electron microscopic structure, and here the polyribosomes consist of mRNA and the polypeptide and its collection on the ribosomes. After release of the polypeptide chain, we have other modifications called post-translational modifications. Some of these modifications we are describing here. First, it folds into biological form and can be further modified to final proteins by one or more of combination of reactions that include hydroxylation, carboxylation, glycosylation, phosphorylations. Cleavage of peptide bonds that converts a larger polypeptide into smaller polypeptide, or a smaller form, and further folding, packing, or folding and packing of protein into its complex configuration. This is another modification. So modification may occur via certain reactions like hydroxylation or phosphorylations, and may occur by change in the size of the polypeptide chain like cleavage, or change in its configuration like folding. Protein folding is a complex process that determined by the sequence of amino acids in the polypeptide chain. In some instances, nascent proteins associated with other proteins called chaperones, which prevent inappropriate contact with other proteins and ensure that the final proper conformation of the nascent protein is reached. Proteins also has information that help to direct them to individual cell compartments. And many proteins are going to be secreted or stored in organelles, and most transmembrane proteins have as their amino terminal a signal peptide, or called leader sequence, which guides them into the endoplasmic reticulum. This signal peptide or leader sequence is made up of from 15 to 30 mainly hydrophobic amino acids, and the signal peptide, once synthesized, bind to the signal recognition particle, which is a complex molecule made up of 6 polypeptides and 7 sRNA. Here again as another form of RNA rather than the 3 common players in the process of protein synthesis. So we have small RNA. It is one of the small RNA. So the signal peptide combined with another structure called signal recognition peptide. This signal recognition peptide is a complex molecule, consists of 6 polypeptides plus 7 sRNA. The signal recognition peptide stops translation until it binds to a translocon. Translocon is a pore in the endoplasmic reticulum, which has a heterotrimeric structure, and the ribosomes also binds, and the signal peptide leads the growing peptide chain into the cavity of the endoplasmic reticulum. The signal peptide is then cleaved from the rest of the peptide by a signal peptidase. Now its role has ended. The role was to guide the polypeptide chain into the cavity, or to pass through the pore of the endoplasmic reticulum and enter into the endoplasmic reticulum. Once this happens, now its function or its role has ended. So it is cleaved and removed from the peptides. The SRPs are not the only signals that help to direct the proteins to their proper place, and there are other signal sequences, post-translational modifications or both, can serve this function of guidance. So this is the growing polypeptide chain, and this is translocon, which is the pore on the endoplasmic reticulum. This is the signal sequence. Once the peptide inside the reticulum, this signal sequence will be removed or will be cleaved. Here, as you can see, it's the color in purple. Here, it is cleaved in this stage as the chain is guided into the cavity of the endoplasmic reticulum. Now we are moving to the process of protein degradation. After finish the process of protein synthesis and post-translational modifications, we will learn about protein degradation. Protein degradation also is well-regulated process and also complex process. Up to 30% of the newly formed proteins are abnormal, and so they have to be removed. They are abnormal. There is, for example, improper folding or any abnormality during the synthesis, and as they have this abnormality, they can't add to the cell, or they can't perform the required function applied to them. Also aging of the proteins. Aged proteins need to be destroyed and degraded. So either the proteins are newly formed but not properly formed, so they have to be degraded, or aged proteins, aging effect on the proteins make them also have to be removed. Conjugation of proteins to the 74 amino acid polypeptide ubiquitin. We have certain protein in the cell called ubiquitin, consists of 47 amino acids. This ubiquitin marks the proteins for degradation. So once these proteins combined with ubiquitin in a process called ubiquitation, now this proteins is marked for degradation. This polypeptide or ubiquitin is highly conserved and is present in species ranging from bacteria to humans. Again, we have a definition of ubiquitation. Ubiquitination is combination of ubiquitin protein to a certain protein, and the result of this combination or the result of this binding is marking this protein for degradation. Sometimes multiple ubiquitin molecules bind to certain protein, and this process is called polyubiquitination. So if single ubiquitin protein combined with the target protein, it's called ubiquitination, but if multiple molecules join the process, it is called polyubiquitination. Ubiquitination of cytoplasmic proteins, including integral proteins of the endoplasmic reticulum, mark them for degradation in certain particles or proteosomes. So this is active protein. Now this is ubiquitin protein. If more than ubiquitin protein, this is a target for degradation. If just one ubiquitin molecule join it, it's called ubiquitination. But if more than one ubiquitin protein, it is called polyubiquitination. In both cases, this protein now is marked or is indicated for being degraded, which occurs in proteosomes. The outcome of the action of proteosomes is cleaving or degradation of the protein into peptides and then into amino acids. Ubiquitination of membrane proteins, on the other hand, such as cross-hormone receptors, also mark them for degradation. However, this can be degraded by lysosomes instead of proteosomes. So the idea is the same. Once ubiquitinated, it has to be degraded. But the site of degradation is the difference between membrane proteins or cytoplasmic proteins. So membrane proteins are degraded in lysosomes, while cytoplasmic proteins are degraded in proteosomes. The rates at which individual proteins are metabolized vary, and the body has a mechanism by which abnormal proteins are recognized and degraded more rapidly than normal body constituents. For example, abnormal hemoglobins are metabolized rapidly in people with congenital hemoglobinopathies. Catabolism of amino acids. The short chain amino acids produced by the short chain fragments produced by amino acids or carbohydrates and fats are very similar in metabolic aspects. They catabolize in a very similar way. And they have common metabolic pool of intermediate. We have common metabolic pool of intermediates from carbohydrates, from proteins, or from lipid metabolism. These fragments can enter citric acid cycle or Krebs cycle, which is a final common pathway of catabolism. In this cycle, there is breakdown of these fragments into hydrogen and carbon dioxide. So this is illustration of citric acid cycle or Krebs cycle. and the net result here we have carbon dioxide, carbon dioxide again. Interconversion, of course, these steps are more biochemistry. Is more than physiological. What is important from the physiological or from the biological point of view is the result of breakdown. It is energy production and breakdown into hydrogen and carbon dioxide. But details are not needed for this course. Interconversion of amino acids, we can synthesize one amino acid from another. So this is interconversion. You convert one type into another. And this occurs by either transfer, removal, or formation of amino groups. Transamination reactions means conversion of one amino acid to the corresponding keto acid with simultaneous conversion of another keto acid into amino acid. For example, here alanine plus alpha ketoglutarate converted, and this reaction is reversible. It can occur in the opposite way. Here it's converted into pyruvate and glutamate. The transaminases involved are also present in the circulation, and when damage or injury occurs to active cells as a result of any pathological process like inflammation, like destruction by cancer, like trauma, serum transaminases level rises, and it is clinically important to measure these serum transaminases levels as an index or as a marker for damage to this organ. For example, the increase in plasma aspartate aminotransferases occurs in myocardial infarction. Myocardial infarction when the coronary blood vessels that supply the muscle of the heart is occluded, so the part of the myocardium or the cardiac muscle supplied by this blood vessel will die and undergoes necrosis. The result of this pathology is increase in the blood level of this enzyme. Also, oxidative deamination of amino acids occurs in the liver, and in this process an amino acid is formed by dehydrogenation, and this compound is hydrolyzed to the corresponding keto acids with the production of ammonium. Very important process called urea formation, in which ammonium formed by deamination of amino acids in the liver is converted to urea, and then urea is excreted in urine. When NH4 plus form carbamyl phosphate, and in mitochondria this carbamyl phosphate is transferred to ornithine, forming citrulline. The enzyme involved in this process is called ornithine carbamyl transferase. Actually, when this enzyme is deficient, genetically absent, or in one of the inborn errors of metabolism, there is increase in urea level, there is increase in ammonia level, because its conversion to the final product or citrulline is hindered, and this patient or this victim may have certain clinical form of encephalopathy or disturbed conscious level. Citrulline is converted to arginine, after which urea is split off and ornithine is generated, and this process is called urea cycle. The overall reaction in urea cycle consumes 3 ATPs and thus require energy. So it is active process that needs ATP. It is not passive process. This is an illustration of urea cycle. Again, biochemical reaction steps are not required for our course. Just have a look on the reactions and on the cycle. You have to know the energy requirement for the process, but the steps and the reactions or the formula are not required for our course. Most of the urea is formed in the liver, so the main site of production of urea is the liver, and in severe liver diseases the blood urea nitrogen falls and the blood ammonia rises. Again, congenital defect or deficiency of ornithine carbamyl can lead to ammonia intoxication, even individuals who are heterozygous for this deficiency. And these patients may have coma or disturbed consciousness level or encephalopathy. Now we are moving to the final part of our today's lecture, which is about to define the main metabolic functions of amino acids. In addition to providing the basic building blocks for proteins, amino acids have other metabolic functions. So in addition to being structural functions, they have different metabolic functions. For example, thyroid hormones, catecholamines, histamine, serotonin, melatonin are and other intermediate in the urea cycle are formed from specific amino acids. Yeah, here you can see thyroid hormones. It's hormones. We have other catecholamines, histamine, serotonin, important neurotransmitters, as well as melatonin. Methionine and cysteine provide the sulfur containing in proteins. The sulfur contained in proteins. CoA, taurine, and other biologically important compounds. Methionine is converted to S-adenosyl methionine, or is abbreviated into SAM, which is the active methylating agent in the synthesis of compounds such as epinephrine. So it is methyl donor. SAM is methyl donor in certain reactions or in production of certain compounds like epinephrine. Thank you for your attention.